The results we obtained in Barcelona within Work Package 5 that deals with monitoring of plankton biodiversity and the evaluation of its links to environmental status. Okay, so biodiversity is one of the main descriptors of the Marine Strategy Framework Directive. And as we've seen here, most of the effort has been put on the monitoring of large organisms that are easily or more easily identified, and they cover a wide range of sizes. But nowadays we know that most of the diversity is contained in the small microbes that form the marine plankton. And these organisms are very diverse, but are also very abundant and relevant for the ecosystem since they form the base of the food web. And nowadays we have the tools to study this diversity. What we don't know is if there's a link to the environmental status of the ecosystem. So coastal waters receive nutrients, pressure in the form of eutrophication, and we know that plankton communities can respond to environmental changes. But what we didn't know is we can link these changes to the assessment of the environmental status. So our idea is that given to ecosystems that are subjected to different pressures, if we study the microbial diversity at different levels, one at the community structure and the second at the indicator species, can we link this information to the environmental status of the ecosystem? So we did this following two different approaches, the two different methodological approaches. One is through flow cytometry, which allows the quantification of major groups of plankton based on their optical properties. It's a high throughput and cheap and easy methodology, and the second is the DNA approach, that it's a little bit more complicated but provides biodiversity monitoring at a higher resolution. So we did our study in the northwestern Mediterranean, in the Catalan coast. It's an area that has been studied through the Water Framework Directive over 20 years, so we have a good understanding of the pressures that this area was receiving from north to south and from inshore to offshore waters. And this allowed us to select the stations to sample, covering a wide range of trophic gradient, going from offshore waters to harbors and through different coastal areas subjected to different natural and agriculture and human pressures. So we analyze the data from these stations based only on the physicochemical data. What we observed that we can distinguish or we can group the stations primarily based on the concentration of ammonium and nitrate and also on the freshwater content. But there is a pool of coastal stations that are subjected to different pressures that we cannot clearly distinguish. If we analyze the same stations, but based on the biological data from flow cytometry, again, it's easy to distinguish the stations based on freshwater content or the very impacted areas, but the coastal areas, we have a gradient that we cannot clearly distinguish based on the flow cytometry data. We look into detail at the organisms that we find. We see that there are some potential groups that could have a indicator of, it could be potentially good indicators because they follow a trend with a trophic gradient, and other groups that are specific for certain types of samples. But from all the different groups that we found, we only found one that could be potentially a good indicator, which is the ratio between two different types of cyanobacteria. But again, this is clearly distinguished the two extremes of station, and it's less clear for the intermediate tones or for the gradient. So this was with flow cytometry. The second step was to analyze the data, the molecular data. And since, as presented here, Work Package 5 had a strong component of validation, we first validated the two most used or available high-throughput sequencing in the market, that is pyrosequencing and Illumina, to see if the sequencing method introduces a bias in the results. And we found a very good correlation, so both at the species, the richness level, and at the community structure, both seem good. So we try to find indicator species or indices of diversity that could provide good information for the assessment of the environmental status. So regarding indicator species or OTUs, which are proxies for species for bacteria, we did find some of them, But again, these were good for distinguishing the extremes, so the very impacted area from the from the least impacted, but less for the gradient. If we look at the indices, we found that in general bacteria seem to be more promising than small eukaryotes, and we found certain indices or between different taxa of bacteria that had a good correlation with the degree of impact that could be potentially good. And the next step was to expand this study to different locations and also to introduce the seasonality, the variability with time. And when we did that, we found that these indices are not so robust. So neither are the richness or the ratio between different taxa. We found that the correlation is not so good when you include the time dimension. And this exemplifies, for example, when stationary is considered good, the variability over 10 years, and one of these indices, despite the trend, seem to be good. When you look into the detail, then it's not as strong. So we could conclude that plankton biodiversity could not be a good indicator, but the problem is more conceptual, that we're trying to find a linear relationship between this pressure and the response in the community, and plankton are fast responders, and they don't integrate the history, the history. And also, another issue is the sampling strategy that we're following, that we can, even if we sample every month, we can see that trend is good, but we don't know what's happening in between, and this can also provide misleading information on the conclusion.